Hello, everybody. Thank you so much for joining our last talk for today. I have the honor to introduce Dr. Ralph Greenspan. Dr. Greenspan has worked on the genetic basis of behavior and brain function in fruit flies almost since the inception of the field, studying with one of its founder, Jeffrey Hall, at Brandeis University in Massachusetts, where he received his PhD in biology in 1979. He is currently the director of the Center of Brain Activity Mapping of the Kavli Institute for Brain and Mind at the University of California, San Diego, and the professor in the Division of Biology's Neurobiology section and the Department of Cognitive Sciences. Most of our current picture of neuronal networks have been built by using single cell recordings. As part of his work with Takeo Katsuki, he wanted to take an alternative approach and conduct a comprehensive network analysis of jellyfish nervous system. They have, this was a side project in the lab that he will introduce to us now. I want to end by saying that with a tip for the happy hour, please ask him about his very interesting history in scientific history and approach to science. And with that, I would like to welcome our last speaker, Ralph Greenspan.
Hello, it's a pleasure to be here and speak to you today. I have to say that I'm not used to Drosophila being the stodgy old guard at meetings, given how fly neurogenetics was viewed when I began in the field. So what I'll describe to you is what has been a three-year fascinating side trip for me from flies into Cnidaria, which was motivated by my realization that to understand the neural basis of behavior, we had to be able to see the entire nervous system in order to be able to chart the interactions in it. And to do that, at the time that I was starting this, it had to be flat enough to see in the single microscope field all at once. And when we tried voltage sensitive dyes, the entire bell of the jellyfish lit up because it's contractile tissue.
If I may interrupt just for a second, your screen is not shared with us yet. I wanted to make sure you were aware of that.
All right, I thought I had done that, but here it is again. So as I said, that was my motivation for undertaking work with the jellyfish, because at the time there was, it's now possible to see the whole nervous system of the fruit fly in one field all at once, which I will describe to you very briefly at the end. But at the time that wasn't possible. So I embarked on this project, and I started out by trying voltage sensitive dyes, but the whole bell is contractile, so the whole bell lit up. So in order to be able to target expression of voltage sensors, I needed genomic sequences. Hence, we began doing the genome. And this is a collaboration among many people over years to get it done. And we published it in Nature Ecology Evolution last year, and that reference is is later on my slides. These are all the people who participated in this. It was spearheaded in my lab by my postdoc Takeo Katsuki, and David Gold from UCLA, who was at UCLA as a graduate student at the time in David Jacobs' lab, played a major role in it from then all the way through to the end. And various others and various other labs around the country. Michael Rogulski from Cold Spring Harbor did some of the initial sequencing. Some of the later sequencing was done in Thomas Holstein's lab in Germany. Rob Steele from UC Irvine also played a role in this. So it's a collaboration among many labs over a long period of time that we finally completed and published last year. And as you all know, what's special about Cnidaria, about this branch of the Cnidarian phylogeny, is the presence of the medusa stage. And so one of the things we were most interested in looking at was what were the genes that seemed to correlate with the appearance of a medusa stage. And I'll give you the result of that when I get to the end of the genomic discussion. So the nervous system of Aurelia is a diffuse nerve net, which in this slide that I have here, this micrograph of it shows it in the ephyra, which was flat enough to see it all at once and small enough to get into a single microscope field. So that was what we initially started trying to do it on. And this was done by Nagiyasu Nakanishi in my lab and various others over the years. So our overall strategy was to sequence the jellyfish genome with the goal in mind of developing molecular genetic tools for jellyfish to use for all the neurobiological goals, like characterizing connectivity of the nervous system, monitoring activity in the nervous system, and ultimately manipulating behavior in the nervous system. We got to the end of the first one of these. The genome sequencing was done in many different labs coordinated by Takeo Katsuki, starting with Illumina paired end sequencing at Cold Spring Harbor and some at UCSD and some at University of Heidelberg and mate pair sequencing there and mate pair sequencing at UCSD and finally PacBio reads at UCSD. And through all of this, we were able to get to the stage of having a publishable genome. And this is a flow chart of all of the aspects of this project. Oops. Where am I? The Illumina portions of it and the PacBio portions of it in a flow chart that finally got us there eventually. As you'll quickly discover through this talk, I'm actually not a genome jock. I've tried to learn it, but I'm not as well versed in it as many of you. In comparison with other cnidarian genomes, the Aurelia genome is not the biggest, not the smallest. At 760 megabases, it's up near the high end compared to all these other cnidarians. It has a repeat content of about 49%, which is slightly less than somewhat less than Hydra, and considerably more than Nematostella and Aptasia. The genome-wide SNP rate is 0.19%, which is 2 to 4 times lower than other cnidarian species, such as Porites, Acropora, Hydra, and Aptasia, comparable to Drosophila at 0.2%, and higher than C. elegans at 0.1 and human at 0.07%. We used a variety of approaches for assembling the genome. We used Trinity for transcriptome assembly. The transcriptome work, I should say, was carried out by David Gold in David Jacobs' lab, starting at UCLA and continuing on into a subsequent postdoc he did, and finally into his own lab. Gene modeling was done by PASA, repeat masking by Repeat Masker and Repeat Scout, gene prediction by GeneMark, Glimmer, HMM, and Augustus, gene mapping by Exonerate and GMAP, gene modeling by Evidence Modeler, the final gene modeling by PASA, and gene annotation by TriNotate. The outcome told us that Aurelia has 34,337 predicted genes, as compared to some 29,000 in Aptasia, 20,000 in Hydra, 18,000 in Nematostella. The RNA-seq data that David Gold produced from the 7 stages of the Aurelia life cycle detected 4,736 differentially expressed genes, which included well-known invertebrate neurosystem genes such as ELIV, RF-amide, GLW-amide, beta-tubulin, alpha-tubulin, beta-actin, and ubiquitin. The interesting thing turned out to be where the new genes were found in the life cycle, because they were not by any means concentrated in the medusa stage. They were evenly distributed all across the many different life cycle forms and stages that it has, suggesting that it was differences in regulation of selected genes that would make the difference. And these genes, if you're curious to see what these are, they can be found by looking in this paper that we published in Nature Ecology Evolution last year. We took our knowledge and went ahead and tried to generate transgenic jellyfish back before it had been done anywhere. And this was spearheaded again by Takeo, my postdoc, Nagayasu, research visiting scientist, Sarah Johnson, my research associate in the lab. After trying and failing to get it to work by electroporation, which merely caused the medusa to break apart into pieces, or chemical transfection, which tended to kill it, or biolistic gene delivery, which blew it to smithereens, we tried doing microinjection of DNA into fertilized eggs, as had worked in Hydra and Nematostella, for which we collected mature medusae, set up stable medusa culture, induced spawning, collected fertilized eggs, and developed a microinjection protocol, which we succeeded in getting to work very well with dextran and also with EGFP mRNA, where we could get expression of fluorescence. And over the course of our 3-year Moore Foundation grant, we were able to increase the efficiency of injection of messenger and mRNA expression up to 25%. Much easier to detect with dextran, whether we've gotten successful injection. And Nagayasu perfected a protocol for single embryo DNA extraction and PCR. Next steps were to have been to use CRISPR to knock out a neuropeptide, as has now been published in Hydractinia, confirm the knockout via PCR, inject an actin GFP plasmid to confirm the ability of an actin promoter to drive expression, then inject that construct via meganuclease for genome integration, and finally do CRISPR integration for targeted genome integration. But alas, we ran out of the Moore Foundation funding, and they don't do renewals for the same project. And we tried to get another renewal from them, but they didn't go for it. In the meantime, since I had my roots in Drosophila, we went back and were now able to develop a technique for performing whole brain optical recording from the fly. This is using the technique of light field microscopy, which allows us to do real-time whole brain imaging faster than confocal and simultaneously all levels of the brain at once, which you then have to deconvolute. That's because they get put through this micro array, this array of micro lenses that distorts the image, and then you can reassemble it, the collected signals from it post-recording computationally. And this is published last year in PLOS Biology, spearheaded by then postdoc in my lab, Sophie Amaw, and Takeo Katsuki, and various others in my lab. Sophie has gone on to a postdoc in Munich in the meantime. These are some examples of recordings from different levels of the brain from that light field microscopy. up at the top here, and it shows it in response to UV flashes and to puffs of ethanol. So the UV flashes give you responses in the optic lobes, correlating with the presence of the stimuli. The ethanol pulses give you antennal lobe responses, as expected, for an olfactory stimulus. And likewise, the mushroom bodies give you responses to the olfactory stimulus, and lateral horn, all known olfactory recipients. And there are various kinds of activities going on in the inferior bridge, in the protocerebral bridge, and in the posterior slope of the protocerebrum, more to the UV flashes than to the ethanol puffs. But I'll show you at the end, later on, some of the activity results of whole brain recording. Here it is. So this is light field microscopy of a fly expressing all through its brain GCamp reporter gene. And this is the whole brain recording. This is the fly actually walking. These are the alternating noduli. This is regions of activity as determined by independent component analysis of a fly turning and walking on a tethered on a sphere. When we used independent component analysis to identify what these regions of activity are, based simply on what are all the cells in a given area that are following the same time course of activity, we were interested to see that they corresponded very closely to the anatomically defined local processing units identified by Anshin Chang in the fly, based on his single cell level mapping and connectivity clustering of the fruit fly brain. And these were activity patterns that were determined in Sophie's paper from Close Biology. And as you can see, they correspond exceedingly well to the areas of anatomically defined local processing units, which are defined as regions in which the neurons start and end within the same region. One of three classes of neurons that he identified as making up the fly-brain connectome, the others being major tracts and hub neurons. These are all of the people who participated in the project over the many years that we did it, many in my lab. As I mentioned, David Gold and David Jacobs from UCLA, Rob Steele from UC Irvine, Thomas Holstein and his student David Iberson from University of Heidelberg, Michael Rogolsky, who initiated the sequencing at Cold Spring Harbor, and a group at UC Santa Barbara in Ken Kosik's lab. Thank you very much for your attention.